Welcome, everyone. I'm Hank Paulson. I'm delighted today to introduce Dr. Joe Abisambra, Associate Professor and Teacher Scholar from the University of Florida, who's giving a special seminar today called De Tau y otros demonios, maladaptive translation in tauopathies. I've known Dr. Abisambra for a number of years. He is a leading expert in the behavior of tau as a protein in disease states. He received his Ph.D. from the University of South Florida and did a postdoctoral fellowship there as well, working, I think, mostly with Chad Dickey and some others, and exploring how tau is handled by various quality control proteins and how quality control proteins play a role. He then went on to University of Kentucky, where he was an assistant professor from 2013 to 2018 and began his own independent laboratory studying mechanisms of tauopathies. Was recruited to University of Florida in 2018 as associate professor, where he is not only associate professor in the Department of Neuroscience in the Center for Translational Research and Neurodegenerative Diseases, but he's also the assistant dean of diversity and health equity. Dr. Abisambra has won a number of awards from both University of Kentucky, University of South Florida, and University of Florida. He is exceptionally well funded from NIH with several R01s, including a new one that explore the relationship between tau production and RNA and protein homeostasis. He also has done some work on special imaging techniques that are involved in neurodegenerative diseases. As you look at his CV, I'm really impressed with the amount of mentoring he has done of junior scholars. Still, I would say a relatively junior scholar himself, he is spending a lot of time helping other scientists become great. And I really appreciate that. Without further ado, I'll turn it over to Dr. Abisambra, who can give us his lecture. Joe.
Thank you so much, Hank. Very kind and generous introduction. And I really appreciate you took the time and did a great job at reading the title of this talk, which I've never really put out there, and I really wanted to take advantage of putting a title in Spanish for many reasons. Some of them may be obvious. I'm originally from Colombia, and so in some ways to give some background to introduce myself personally, but also to honor one of our best and best known authors, Gabriel Garcia Marquez, who wrote Of Love and Other Demons. And he was one of the best recognized authors, because in 1985, he won the Nobel Prize for writing 100 Years of Solitude, Cien Años de Soledad. And I wanted to honor Garcia Marquez because later in the late stages of his life, he developed mild cognitive impairment. And as we all know, especially for this audience, that in many cases, MCI may develop then into later stages dementia and or neurocognitive disorder. And as we all know, dementia is a decline in mentation or mental activity. And it may lead to changes in personality, social behavior, and all of these symptoms that present. The most common form of dementia are tauopathies. And the most common tauopathy is Alzheimer's disease, which you're all very familiar with. And in line again with literature, here we have two of the first patients ever described to have dementia. And on the left, we have Don Quixote de la Mancha, who's described in 1605, and then King Lear, sometime around that time. There's some interesting and completely irrelevant parallels between these two. Shakespeare and Miguel de Cervantes Saavedra were two of the most important authors in Spanish and English. And not only did they describe these two individuals at the same time, they also died the same day. Very weird and completely unrelated, but I wanted to share that. Going back to tauopathies, we've been very interested in them. They are more than 20 different neurodegenerative disorders that share a common pathological hallmark, which is the intracellular aggregation of tau. And we see this in very different forms or faces of tau aggregates. in Alzheimer's, corticobasal degeneration, progressive supranuclear palsy, and even chronic traumatic encephalopathy. We see aggregation of tau, not only in neurons, but other cell types, and showing different types of aggregates. So the overall objective of my lab is to establish the molecular mechanisms that link tau abnormalities and tau pathology with brain dysfunction. And in doing so, we may identify novel therapeutic targets, establish molecular mechanisms of normal brain function to better understand tau biology and understand how the brain may work, or the brain works, and finally to target fundamental processes, and in doing so, modify disease outcomes. Our current research focus is based on a number of individuals who are really the backbone of the lab, and they have built a fantastic environment to work in and a very fun environment, and they are brilliant, which has led us to build 3 different branches of research. One is on traumatic brain injury, in which we're focusing currently on a model of repetitive mild TBI, leading to concussions, concussion syndrome, and that type of model. We're trying to identify effective biomarkers and test interventions. The second branch, which will be the focus of my talk today, has to do with the maladaptive translation. And I'm going to focus on the interaction of tau with RNA and ribosome complexes, as well as touch on the unfolded protein response, and in particular kinase called PERK. The third branch, which has given us a lot of freedom to look into a lot of fun and high-risk projects, has to do with looking at muscle-brain interactions. We've done some work in this space with collaborators in the University of Rio, also neuronal cilia, and we're developing new approaches with multiphoton microscopy to do live imaging in mice that are awake. And this is in its early stages, but we're getting into that. Just to share with you what we're doing in the space of TBI, we've developed a platform with not only the objective of following our own models to test hypotheses, but also to build collaborative efforts. So for example, we now have 2 models of injury called Chimera, one for mice and rats. Mice are injured, and this model is a close head injury model of engineered rotational acceleration. Essentially, the mice are hit at the top of the head. They're laying on this platform. The piston hits them, and it develops this rotation. It is truly remarkable that we observe a reproducible type of damage, particularly in white matter. Mice don't have a whole lot of white matter, and we can see time and time again the reproducible immunohistochemical damage and inflammation markers and disruption of white matter tracts. From here, we can get our functional outcomes, both in behavior, and we're doing a lot of work using the Amherst facility here at UF with our 11T MRI system, where we can place nodes and measure functional connectivity with these node strength maps. Since this model presents a unique and reproducible type of damage in white matter tracts, it also presents a challenge in that we can't really isolate the whole brain, homogenize it, and do other types of protein measures. So we've partnered up with our colleague next door, Dr. Stefan Prokop, who acquired a nanostring instrument, and we can do geomics digital spatial profiling. And as you can see here in this recording, what we can do is identify specific regions of interest. Here I'm highlighting the optic tract, and get different profiles of proteins, whether they were from injured mice or not, or the sham. And so we're using this platform to investigate the effects of aging on head injury, because early injuries in early life seem to lead to increased risk for dementia over time. We're testing this in different tau models to see what are the mechanisms linking TBI to tau pathology. We're testing new therapeutics with collaborations, particularly with Luke Wiseman at Scripps. And as I mentioned, biomarkers and molecular mechanisms. Then in terms of the multiphoton microscopy, I just wanted to share with you that this is the system we have. And here we're working with Todd Goldie, who's developed a pipeline of organotypic cultures where they're creating or culturing whole hemispheres are down to 400 micron slices. And we can now put them, after injecting with different types of viruses, coding for fluorescent proteins. And in our multiphoton microscope, we can get outstanding resolution of what you can see here are a series of tangles. And I'm really excited about this because we can now take this type of imaging back to a live mouse and awake doing some type of behavioral task. And with this model, we can now study stroke, memory, blood-brain barrier integrity. For my personal interest, translation in situ in real time. And we're getting into the area of sleep disorders. And again, this is a collaborative platform with other folks here at UF. But today, as I mentioned, I'm going to talk to you primarily about maladaptive translation on the basis that while tau is best known as a microtubule stabilizing protein, under certain conditions that are still unknown, pathological tau species impair translation in direct ways by interacting with RNA and ribosome proteins, or indirectly by activating, for example, the unfolded protein response and this kinase called PERK. So getting a little bit into the history of tau, was described in the late 70s as a major microtubule binding protein. And this was a really cool time because they were just starting to investigate tau at a basic science level. However, when it was described as a component of tangles in the late 80s, the field of tauopathies, and in particular Alzheimer's disease, really got into studying how tau may lead to or link to pathology and dementia. And the basic biology of tau was not at the forefront of the field. So what we know since then is that tau is best known as a microtubule stabilizing protein, as I mentioned, and it is heavily post-translationally modified, which makes it a fascinating protein because not only is it heavily phosphorylated in all these different sites, it is also subjected to splicing, leading to 6 tau isoforms in the adult human brain. There are various areas that are acetylated, methylated, nitrated, ubiquitinated, acetylated, methylated, and cleaved, leading to different fragments of tau that may play important roles in disease. In general terms, tau so far has taken a pretty bad reputation from my talk, but it does play a critical role for the cell. While neurons inarguably are beautiful cells, they do have these extended prolongations which add to their complexity. And that presents also a challenge. For example, we need transport of cargo from one side of the cell to the other. For this to occur, we have, as an example, a mitochondrion that needs to be transported from the soma down to the axon. And for this, we have alpha and beta tubulin forming polymers. And interspersed between the polymers, tau stabilizes these stretches, leading to the formation of a mature microtubule. Then, thanks to motor proteins, this mitochondrion can now be transported from one side of the cell to the other and serve whatever function it may do at distal areas of the cell. But given that the intracellular environment of a neuron is highly crowded, this dynamic microtubule must be disassembled. And in this process, tau is phosphorylated, reducing its affinity for the microtubule and now allowing for the microtubule to disassemble. Now, I recently heard from a talk that tau is not really an intrinsically disordered protein. However, tau is biochemically described as an intrinsically disordered protein, and I take it as an important and really a complement to this complex protein, because it has these areas that are not stable. The tau is a very hydrophilic protein that can aggregate, and part of it is because it has all of these important regions that are either hydrophobic in the middle of it, and it has this stretch of proline-rich region that allows it to twist in many different directions. So under some of these conditions, tau can be soluble And under abnormal conformations of the soluble tau, it can lead to oligomers being some of the most toxic conformers that alter neuronal and cellular function in general. Under pathological conditions, even though those mechanisms remain unknown, what we do observe postmortem is that tau is hyperphosphorylated, and as disease progresses, tau forms these aggregates surrounding the nucleus. Now, there are evidence showing that tau does not strictly associate with microtubules. In fact, there are numerous binding partners that may give us more insight into what is tau's true function, or how does it participate in normal brain physiology. And we did some of this work back in 2013. In fact, this was some of the last experiments that I did as a postdoc in Chad Dickey's lab, where we identified that tau associates with ER proteins in the endoplasmic reticulum. And so the endoplasmic reticulum became a central organelle, not for my work, because it is a central organelle to the cell. It connects with the nucleus by facilitating translation. It also has its own system for proteins to fold properly. It has its own chaperone system. Proteins from here, once folded properly, can go to the Golgi apparatus or the plasma membrane. If they don't fold properly, they can be degraded through ER-associated degradation to the proteasome or autophagy. If the ER undergoes any kind of stress, it'll activate its own unfolded protein response system to cope with that ER stress. It connects to mitochondria, it connects to calcium homeostasis, and it also participates in lipid and carbohydrate metabolism. What we found in that paper in 2013 is that tau associates with the ER. And so in many ways, we started thinking that, well, it impairs ribosomal ER function. But what does that mean to the cell? What pathways are affected? So we continued on with some of this work in my lab at Kentucky, first by validating our early results and showing here, for example, that the PHF1 positive, this is an antibody that we obtained from Dr. Peter Davies, recently passed away. And it identifies phosphoserine 396, serine 404, and is characteristic of later stage tangles. And here we're showing that it overlaps in staining. In fact, there's partial colocalization with an ER protein called calnexin. Again, showing that tau and PHF1 pathological tau species partially colocalize with the ER. So to better understand what is present in these tau-associated ER proteins, what are they composed of, we began our work doing subcellular fractionations and mass spectrometry to identify those proteins. And here we're showing how we enriched for ER proteins simply from human brains. Now, one of the advantages that I had at the University of Kentucky was access from Pete Nelson to a wonderful brain bank where we had the privilege of using those brains for these experiments, and we communoprecipitated tau or actin as a control from human Alzheimer's disease brains and non-demented age MASH controls. Ran them on gels, cut the bands, and did mass spectrometry. And we did this in 2 different experiments at 2 different times. One with a tau-5 antibody, which identifies the middle region of tau. And then this was an important antibody that identifies the carboxy terminus, because we didn't want to pull down newly transcribed tau. We wanted to pick up the tau that's already in the cell and perhaps undergoing some pathological process. And we were very surprised to find that a large component of the proteins that associated with tau, both under normal and Alzheimer's disease brains, participated in translation. And many of them were very different between the Alzheimer's and our controls. So this tau ribosome association had been described for more than 30 years. And one of the first groups to describe this was actually Skip Binder's lab in 1987, where using electron microscopy techniques in human Alzheimer's brains, they found that tau actually decorated ribosomes in dendritic branching points. Although these phenomena had been described, it wasn't until a few years ago that we described what happened. What are the consequences of these interactions? And so in the next couple of slides, I'm going to show you the work that we published in this regard using 3 in vitro models. First, we did some experiments in a cell-free assay, then using an immortalized cell line that we called inducible HEK cells, and finally, using primary neurons. Our hypothesis is that under normal conditions, in neurons, tau associates with ribosomes and anchoring it to microtubules, allowing for translation to occur in any particular place along the microtubule. But under pathological conditions, toxic tau species associated with the ER and may even block the mechanism by which new proteins are made. And in doing so, there's a reduction in translation. So to test this hypothesis, we began by going back to our subcellular fractionation and obtaining 2 different fractions of interest. One was a fraction that was devoid of ribosomes and another one that was enriched. And we compared it to the whole cell lysator input. And here I'm showing you the results of a Western blot using these 3 subcellular fractions, one containing the control and the Alzheimer's brain. I don't have to explain much other than to show that in the fraction that only had ribosomes, there's a dramatic increase in tau in the Alzheimer's brains. So tau seemed to be enriched in the fraction that also contained ribosomes. And much of these higher molecular weight species corresponded to the pathological tau, PHF1 positive tau. Here I'm showing 2 ribosomal proteins, P0 and L28, showing that our enrichment was effective in the fraction that only had ribosomes. In HSP70, which is primarily a cytoplasmic protein, was not present in the fraction that only had ribosomes. Using immunofluorescence staining of a human Alzheimer's brain here, a representative of the work that we did in that paper as well, shows that pathological tau species, in this case tau oligomers, which are emerging evidence shows that it's one of the most toxic tau conformers, overlaps in staining with this ribosomal protein S6. But again, what are the consequences of the tau ribosome association? So we began our experiments here to answer this question using a cell-free assay. In this case, we had a 96-well plate and used in each well all the machinery necessary for translation to occur. That is initiation factors, elongation factors, ribosomes, tRNA, et cetera. And we included GFP RNA to promote, to be able to see translation over time in real time over the course of 6 hours. And in different wells, we added different proteins to test. When we added tau oligomers compared to BSA controls, we observed that GFP levels were decreased by about 40% over the course of these 6 hours. These data show that this is GFP again, and suggest that when tau oligomers are present, there's a reduction in translation. Then to test whether this was the result of tau specifically or the oligomeric conformation of any protein, we did the experiment, repeated the experiment using insulin oligomers or even alpha-synuclein oligomers, and we found no significant difference in GFP levels compared to BSA controls. So our data suggests that tau oligomers are sufficient to reduce translation in an in vitro setting, that is in a cell-free assay. Taking this now to an immortalized cell line, we, in a collaboration with Chad again, we requested some of these inducible HEK cells. We developed them in his lab when I was a postdoc, and we generated a line that was inducible. That is to say that when we added tetracycline to the cells, we turned tau expression on. And here you can see over the course of 4 days, tau levels are expressed. And we paired this with the technique called sunset. This is surface sensing of translation. In this assay, we're taking advantage of the fact that puromyosin is structurally analogous to tRNA. So when we add it to the media, it incorporates into ribosomes and fooling them into thinking that it's tRNA. So any new protein that's made will now have a puromycin tag. And thanks to anti-puromycin antibodies, we can now detect the amount of new proteins being made and the distribution. So here I'm showing data indicating that when we turn tau on over the course of 3 days, we see not only the total tau levels increase, but also PHF1, pathological tau. And as PHF1 levels increase, puromycin, again showing the amount of proteins that are made, decreases over time. So again, as pathological tau increase, protein synthesis decreases, or the levels of new protein are decreased. Then we did a rescue experiment. Here we turned tau on, expression on for 24 hours or 96 hours, and we can see the expected increase in PHF1 and decrease in puromycin. But then when we washed off the media after 4 days of having tau turned on and left it for 1 day or another 4 days, we see that PHF1 levels respond to the reduction in tetracycline, and we get complete restoration or even more protein synthesis. So I've shown you the effects in a cell-free assay and an immortalized cell line of what happens to translation when tau is overexpressed. But to make this more neuro-relevant, we went to a model of primary neurons, and we obtained these mice from Dr. Jada Lewis, who co-invented the mice. These RTG4510 overexpressed P301L human mutant tau, which is associated with frontotemporal dementia. There are some recent papers showing that there are concerns with the root cause of brain atrophy in these mice. But for our purposes in primary neurons, the model serves to answer what happens to some of these, to the translation of specific proteins involved in neuronal function. So here in panel G, I'm showing how puromycin levels are again decreased in tau transgenic mice in the 4510s. And here we have total tau increased. One of the proteins that is reduced that we looked at was PSD95, a protein involved in, it's a postsynaptic density protein. To quantify this Western blot, we see again that there is about a 40% decrease in total protein as measured by puromycin, total new protein, and PSD95 levels are decimated in the 4510 primary neurons. But we're not sure whether this is an effective transcription or translation. So we did RT-PCR for PSD95, and we see that RNA for PSD95 is actually increased in the 4510s. So we see a lot more RNA and less protein, suggesting that there's a problem with translation in the 4510s. So again, to summarize this, I've shown you evidence that in 3 in vitro models, tau impairs or reduces translation. But we were not sure whether this happened in vivo. So here to summarize that, pathological tau species in our hypothesis was that it reduced protein synthesis. But whether this happens in vivo or not is unclear. And I'm going to show you evidence now with the 4510s that there is a reduction in a pool of proteins in the translation of specific proteins, but there is also a shift in which other proteins are increased. One of the advantages of the 4510 mice is that Jada was very astute in placing a TET off system in these mice. So here, when we feed the mice tetracycline, tau expression is turned off. And here we did sunset to test what happens to translation in the brain when these mice are overexpressing tau. And here I'm showing you some experiments that a very talented graduate student in my lab, who is now doing a postdoc at Vanderbilt, she injected puromycin intraperitoneally, entered the brain, and here you can see in the dark staining that it stained new proteins. At 5 months, there's a reduction that is not significantly different. But at 7 months, there is. And this is important because in the timeline of disease in these mice, by 5 months, RTG4510s already have cognitive impairment. So if the suppression of translation is a contributor to cognitive impairment in the mice or any kind of abnormalities, it would not be present, it would not be important at 5 months. Maybe it'd be a late stage thing. So we hypothesized that maybe the effect is not that all translation is reduced, rather a subset of proteins. So to test this hypothesis, we treated mice with doxycycline between 3.5 and 5 months old. So here we have our RTG4510 mice. They were expressing human mutant tau. You see progressive tau pathology starting at 2 months, cognitive impairment. There's some early reports that showing that there's mild impairment sometime between 2.5 months, and that progresses. There's neuronal plasticity. We have some evidence that there's damage of LTP at 3 months that progresses and becomes much more marked at 4 months. There's marked neuronal loss at 5 months. So we treated with doxycycline before there's substantial brain atrophy. And this is important because whatever effects we wanted to measure, we wanted them to be independent of the brain atrophy, perhaps even to give us an idea of what's happening during the progression and early stages of the disease. At this stage, we injected the mice with puromycin and took half the brain for microarrays to measure what happened to RNA, and the contralateral side of the brain for IP of puromycin, and then do proteomics based identification of the translatome. This is to show that when we suppressed tau, when we treated tau mice or fed them doxycycline, tau expression went down by about 50%. Here are the data with the microarrays. We found 3 distinct patterns. One pattern was that of mice, the RNAs that were rescued by the reduction in tau. That is to say, in our non-transgenics, they were low, or these were the normal levels. Then in the transgenic mice, we see a massive change in the expression of these RNAs. But when tau was suppressed by treating with doxycycline, we saw a reduction in the transcripts. This is a signature of RNAs that were resistant to the reduction of tau. This is an important control, which is what is the effect of doxycycline? Not surprisingly, mice treated with doxycycline had a different expression of these RNAs, and these many of these correspond to proteins involved in inflammation. But given that we're very interested in ribosomal proteins and translation, we focused on transcripts coding for these proteins, and we found that there was no significant difference in the amount of RNA coding for these proteins in mice that were either transgenic or treated with doxycycline. So that means that in our mass spectrometry, whatever changes we could see in puromycin tagged proteins, that is to say the translatome, we would find that they would be the direct response of translation, not necessarily because of transcription. And here are the results of our proteomics. We see that in, again, the puromycin IP, liquid chromatography tandem mass spec. There was no difference in the proteins between vehicle-treated and doxycycline-treated non-transgenics. Proteins involved in translation were reduced. Here I have some gene ontology classes, and those involved in translation and ribosomal protein production were relatively reduced compared to controls. But when we treated with doxycycline, we saw a rescue in the levels of these proteins. These data suggest that when tau is expressed, show that when tau is expressed and in early stages of pathology, we see a reduction in the types of proteins that are involved, particularly those involved in translation. So then we investigated, well, is this reduction or shift in translation because of tau interacting with ribosomal proteins? One of the ribosomal proteins that we became very interested in, and we had published some of the work showing that tau interacts with this particular protein, S6. S6 is an important protein that regulates translation, and it allows for selective translation of unique transcripts. This protein is S6. It is activated by phosphorylation. It is downstream of the mTOR pathway, and it promotes translation of proteins involved in cell growth. These proteins specifically are called 5' terminal oligopyrimidines, 5' top mRNAs, and they code for proteins involved in initiation, elongation, and all structural ribosomal proteins. So when S6 is phosphorylated, we see a ramp up in the production of 5' top mRNAs, allowing for more machinery for cell growth, more translation to occur. And here we're showing some data in our inducible HEK cells, once again, showing that when tau is turned on for 1 day or 4 days, we see a substantial accumulation of total tau and pathological tau that increases over time. By the course of 4 days, when pathological tau levels are the highest, we see virtually no phospho-S6 levels, suggesting that, again, when tau levels are increased, phospho-S6 is decreased, and therefore there's less likelihood of ribosome machinery production occurring at this time. I showed you earlier this figure indicating that tau oligomers colocalize with this ribosomal protein S6, which was serendipitous at that time, picking that particular antibody. And here we followed up with some immunoprecipitation assays from human non-demented controls and late-stage Alzheimer's disease brains. When we pull down S6 or tau, the reciprocal Western blot shows that both proteins interact. And this interaction seems to be more robust in the Alzheimer's disease brains. But we don't know, once again, if this is a response of the consequence of transcription and not translation. So here we have some RT-PCR for a handful of 5' top mRNAs, RPS6 being one of them. We also had EIF3E, RPL28, and EIF2S1. And here we can see that there is an accumulation of these RNAs or an increase in early stages of Alzheimer's, but it is significantly increased in late stages of Alzheimer's disease. And while the RNA is increased, we see here in this Western blot that the proteins are significantly decreased. So once again, we have more RNA and less protein, suggesting that in the human brains, there's also an impairment of translation. From here on out, and currently, we're working on characterizing more nuances of this interaction. Because our goal then would be, if we could identify the region of the ribosome that is responsible for binding to tau, then a peptidomimetic could break apart that interaction and interfere with whatever negative consequences of this pathological tau species may have on ribosome function. These data show that the proline rich region is essential for the association. Here we have flag tag tau overexpressed in cells. When we did an immunoprecipitation, it was only in the areas that lacked the proline-rich region, either the first or the second domain or both. The ribosomal proteins S6 and RPL19 did not co-immunoprecipitate with tau. So we have this tau-RNA interaction. Now, I've worked with Dr. Ben Wolosin in a few collaborations before, and his lab has been very focused on investigating the formation of stress granules in different models of tauopathy. And that has to do very much with tau RNA interactions. So we investigated whether we could also observe in some of our models and in exploring this association of tau and RNA and how that may impact translation. One of the challenges that we have with the models used in our lab is that whether it's mice or iPSCs, or our immortalized cell lines, is that we're overexpressing tau. And I've always been concerned with the potential artifacts of that interaction simply by increasing the levels of tau. So for this experiment, we went to Dr. Prokop next door and Axis Brain, once again, with the privilege of using those tissues from our brain bank to perform enhanced cross-linking immunoprecipitation. For this assay, we're essentially cross-linking RNAs to all proteins and immunoprecipitating tau, and then doing RNA-seq to identify the types of RNAs that associate with tau. For this, we use 3 different types of brains: early, mid, and late stages of Alzheimer's by Brock. I can tell you that the early AD classification may very well be PART, which is primary age-related tauopathy. One of the most important findings here is that, once again, Alzheimer's brains, in those conditions, we see that there's a vast amount, a very robust association of RNA that co-immunoprecipitated with tau. Of the RNA that pull down with tau, in the moderate stages of disease, 40% of it corresponded to coding sequences, but 43% corresponded to proximal introns, areas that are not described or attributed in any kind of coding sequence. And there's a shift over time in severe cases of Alzheimer's, where now we have different types of RNAs, sequences that associate with tau. So we have this interesting finding that tau may associate with non-coding RNAs. Here are some of the consensus sequences that associated with tau in different areas. I've talked to Sami Barmada about a year ago, and again today, discussing the potential identity of these sequences and what roles they may play. Here we're looking at some areas that participate or are important for splicing and initiation starts. There are some AUG sequences here, as well as GU areas that, enriched areas that seem to participate or have play some role in RNA stability. But I wanted to share this with you. None of this is published, of course, that showing that some of the RNAs that associated with tau caught our attention are SNORDs, small nucleolar RNAs, and they participate in splicing, in the formation of RNA polymerase II complexes and their association with DNA for transcription. And many of them are increased, their associations increase with tau in Alzheimer's, decreased, and others are increased in Alzheimer's disease. So we're getting a sense in the big picture that this association of tau with RNAs under normal conditions could play a role in managing the types of RNAs that are going to be translated and how they're spliced, for example, or processed. And under disease conditions such as Alzheimer's, there are shifts in the types of these RNAs that associate with tau, and therefore that may contribute to differences under a stress condition such as disease for allowing proper RNA processing. But ultimately, can we rescue translation and improve these outcomes? While overall translation is shifted and we can't really encompass the vast amount of RNAs that may be impaired in how they're translated into protein, we do know from some of the work that I did early on that there's this kinase called PERK that's involved in tauopathy called progressive supranuclear palsy, PSP. The summary of that paper in 2013 is represented here in the schematic. So here we have a horrible rendition of a cell. Then we have our nucleus, the endoplasmic reticulum. Here I'm showing some ribosomes for the rough ER. Here's PERK, a transmembrane protein, and I'm highlighting the proteasome. Under normal conditions, well, the ER offers an optimal environment for proteins, nascent proteins, to fold properly. In this case, we have a protein that has folded properly and will be performing whatever function as a transmembrane protein. But not every protein will share the same fate. Here I have this fragment that did not fold properly, and then it will be partially translocated outside of the membrane. It becomes ubiquitinated, and it gets destroyed through ER-associated degradation, where proteins such as VCP or P97 identify the ubiquitinated substrate and take it to the proteasome for degradation. What we found was that tau impairs ERAD, and in doing so, there's an accumulation of ubiquitinated proteins surrounding the ER. This accumulation leads to ER stress. Under these conditions, there's an adaptive response called the unfolded protein response, and that's in the early stages. It helps survive. In this case, here I'm showing that the kinase PERK dimerizes, autophosphorylates, and reduces translation by phosphorylating the subunit EIF2 alpha, or the eukaryotic initiation factor. So in other words, here we have the unfolded protein response in early stages. As I mentioned, it's adaptive. The UPR has 3 branches, one through IRE1, one through PERK, ATF6, All I want to highlight here is that during this phase, we have every green box or circle here showing that there is an acute, in the case of PERC, attenuation of translation to reduce the input of proteins into the ER. But if this pathway is active for too long, these other branches, IRE1, leads to the formation or production of proapoptotic proteins. And in cells such as neurons, which are highly sensitive to protein synthesis for their function to be optimal, if you have chronic attenuation of translation, then there will be a reduction in synaptic proteins. PERC is also genetically associated with tauopathies. Here in 2012, I'm showing that Jerry Schellenberg, in a massive effort, identified a SNP on the gene coding for PERC that's associated with PSP. And pathologically, PERC is also associated with tauopathies. Here I'm showing that in frontotemporal lobar degeneration brains and Alzheimer's, there's an accumulation of the active form of PERC, or phospho-PERC. So this kinase PERC is associated with tauopathies, both molecularly, pathologically, and genetically. So we focused on PERC because of its job, which is to attenuate translation. Here we have an adaptive response where phospho-PERC phosphorylates its substrate, EAF2 alpha, acutely reduces translation, and that allows for survival. But late stage, some transcription factors such as ATF4 facilitate the production of a proapoptotic protein called CHOP, and it leads to cell death. PERC has been associated with other conditions, neurodegenerative conditions, and I've had the good fortune of collaborating with folks in the University of Rome and Sapienza University, looking at Down syndrome. We've done some work in traumatic brain injury. We have some preliminary data in spinal cord injury, and again, some of the work in our models of tauopathy. So the idea is that pathological tau species induce ER stress, activate PERC, and that leads to neuronal dysfunction by attenuating translation long-term and activating proapoptotic proteins. So I hypothesized that if we could block PERC, then we could reduce that neurotoxicity, also increasing translation. So here I want to mention that this project was partially funded by GlaxoSmithKline, who also contributed to the project by giving us copious amounts of this compound, GSK260614, which is a PERC inhibitor. So if the question was, can we rescue translation and improve outcomes? And to summarize, PERC is a UPR kinase, and since tau activates PERC, we should be able to inhibit it with GSK2614 at the right dosage. So here I'm showing that after only 2 days of treatment, oral gavage of the compound, of the PERC inhibitor, in 8-month-old RTG4510 mice, which already have a reduction in translation, we were able to rescue the pure myosin-elated proteins, that is the nascent proteins. So we know that the drug works. And this is our study design. We have our RTG4510. Once again, the timeline showing expression, pathology, cognition, deficits in plasticity, neuronal loss. And we had shown, along with Giovanna Malucci's work, that PERC levels are expressed beyond 6 months. So we inhibited PERC early on. And our initial study was to inhibit PERC for 3 months. But we found after only 1 month, when we did volumetric MRIs, that atrophy was prevented in the transgenic mice treated with the 414 compound. So treating with a PERC inhibitor before there's brain atrophy actually protected the brain from neuronal loss. So then we used, again, this MRI approach to do a functional evaluation, primarily based on calcium. So here we did Manganese Enhanced Mri with R1 mapping. This is a technique developed by Alan Koretsky over 15 years ago. And it's based on the fact that divalent manganese enters voltage-gated calcium channels. Calcium influx into the cytoplasm is necessary for neurotransmitter release. Manganese can enter these voltage-gated calcium channels and replace calcium. And since manganese is paramagnetic, we can detect it with MRIs. This is what it looks like when we inject manganese chloride. After 6 hours in our R1 mapping, we can detect these types of images and quantify them. And here I'm showing the data with non-transgenic mice in panels A and B, and the transgenic mice in panels C and D. Here we're showing that the drug didn't really change much, and the quantification of the delta R1 in our non-transgenics didn't really show much of a difference in the dentate gyrus or CA1 regions of the hippocampus. But here you can see anatomically, there's thinning of the hippocampus. Here's brain atrophy represented by the massive hole that's in the ventricle. This in blue is CSF, and all of this phenotype is protected by the drug in the RTG4510 mice treated with 414. We also did other functional readouts such as open field. Here the red lines represent the tracking of where the mouse ran. When the mouse is in the periphery, it represents an anxiety-like behavior, which means that it's protecting itself from a predator, or that's what it's thought to be, how it's interpreted. When the RTG4510 mice were treated with the drug, this mouse in particular didn't seem to care much where it was running. It seemed to be anywhere. So it seemed to have reduced this anxiety phenotype that is typical of the tau transgenic mice. We also did Y-maze, and we found, again, that when the transgenic mice were treated with vehicle, there's this massive deficit in the entries in the novel arms, but that deficit is rescued by the compound. To get at mechanism, we tried to establish or investigate whether this was a response of tau reduction, and 414 did not reduce tau or phospho-tau. So hunting for mechanism, well, it's an ATPase inhibitor. So we started looking at other kinases that it may inhibit, and certainly there are over 200 proteins that are changed, not specifically due to PERK. And to identify them, we did some mass spectrometry. In this case, we did tandem mass tagging, and we found many, many proteins that were changed. Here I wanted to show you that tau was one of those that was not changed in the transgenics, whether treated with inhibitor or vehicle. So to hint at mechanism and go after this, because it is a powerful functional rescue, we looked at different pathways. Indeed, translation is rescued. We see an increase in puromyosin in the transgenics treated with 414. We saw nitroxidative damage was reduced. Here we're looking at 3NT, which is, so to speak, locally a marker of protein scarring, protein damage, and it's reduced in the transgenic mice treated with 414. Not every pathway was affected. Here, for example, we're looking at lipid peroxidation. That was not changed. But we did find 4 different proteins that changed. And this was identified looking at work from Nick Seyfried in 2 different papers where he identified, we basically correlated all of the 186 proteins that we found that were changed by the drug, either up or down in the transgenics, to those that are changed in Alzheimer's compared to controls and Parkinson's. And we narrowed it down to these 4 proteins: PAXN1, GJA1, BSNL1, and NDUFB5. And we're currently looking at how these proteins change. So to summarize, I've shown you data in the first part of the talk that when tau associates with RNA and ribosome complexes, a lot of these occur surrounding the endoplasmic reticulum, and tau may interfere with their function. It occurs in Alzheimer's disease, and the ribosomal protein S6 may play an important role in this process, either pathogenically or during the progression of the disease. We're currently studying these interactions in much more detail. Tau associates robustly with these RNAs in Alzheimer's, so also the tau RNA, independent of ribosomes, is an important topic to us. And finally, I wanted to label this more as a broad spectrum kinase inhibitor, that while it inhibits PERK, it also inhibits other kinases. And this broad spectrum kinase inhibitor, although it didn't affect phospho-tau levels, it certainly conferred benefits to this tau transgenic model. And it's important right now to, for our work, to establish which pathways are important, because they are potentially clinically relevant. So with that, I want to acknowledge the lab, particularly Shelby Meyer, who did the most of the work in the early part of this talk from the University of Kentucky, and our lab in Florida, currently composed of really talented individuals. I'm very grateful to participate and continue to develop our work. I want to thank Chad, who passed away a few years ago, tragically. He inspired most of the work that I presented today. My collaborators at the University of Florida, from Jada with MI, Stefan, who gave us the brains, Todd, Brenda, Karen, Dave Borscheld, Matt Lavoie, who we recently recruited, Eric and Kotaro, who are helping us with the RNA ribosome studies, Marcella, who is currently helping us with the MRI, Karen Esser with some of the work with the muscle brain that I mentioned, and Jeremy McIntyre with the neural cilia. Folks in the University of Kentucky who have helped us develop not only our studies in human brains, but also TBI. Peter Davies with his antibodies, Rakesh Kayed antibodies, and recombinant proteins. Sapienza University, and our help at the clinical level, the treatment therapies with Luke Wiseman. Our funding from NIH, all the previous funding that helped with much of this work, and our current and future with NIH, Alzheimer's disease, Bright Focus, Rotary Club, and others. And with that, I want to thank you very much for your attention. I hope we have time for a few questions at least. Sorry.
Thank you, Dr. Abisambra. I really enjoyed your presentation. We do have a little bit of time for questions, and feel free to chime in if you have a question. I guess I will just start off by leading very quickly, Joe. You have this arm of work on traumatic brain injury, and you did allude to the fact that the stress response is altered in TBI. Have you been looking at sort of the dynamics, the kinetics, the relationship between phosphorylation of tau in your traumatic injury model and translation and integrated stress response?
Great question. We did back in Kentucky using the controlled cortical impact model. There is and this is all preliminary, so I don't want to put too much on it, but we have found a very puzzling effect, which is an increase in protein synthesis, primarily in neurons after head injury. And we do see an increase in phospho-PERK. We see an increase in other ER stress markers, but it is puzzling because the main marker of ER stress is not changed. So I'll summarize that with the fact that this is a very complicated pathway. Injury certainly plays a role, and we're investigating the time at which it's active and the long-term consequences of translation in the unfolded protein response. And you pointed out something important, which is the integrated stress response, which is another part of the work that we're doing.
Thank you.
Sure.
Other questions for Dr. Abisambra? I know some people have to leave. Frankly, I have to chair a session in a few minutes myself, but are there other questions that people have for Joe?
Yeah, I'm sorry I went.
You did not go too long. It was perfect.
I'm happy to answer emails or reach out to me if there's an opportunity or any questions. Thank you.
Yeah, well, thank you so much again, and we appreciate it. Again, this talk has been recorded. You can go back and look at it. We will make sure that others who couldn't make it today have a chance to look at it. Feel free to reach out directly to Dr. Abisambra if you have questions about the work, which is fascinating and is highly impactful for so many tauopathies. Thank you once again for coming today.
Thank you.